The next unit is repro and development, and we'll start with the male anatomy. You'll see that this lecture has a lot of, well, as we get into it, a lot of pictures from the atlas that I require. This picture is just showing you the development of the external genitalia at about 5 weeks development. And there's some things that are color-coded. So when it's in the indifferent stage, I mean, it's genetically determined, but it'll develop one way or the other. But let's kind of know the genital tubercle. The urethral fold in pink. As well as labial scrotal swelling. If it's the male pathway, the male has inherited the Y chromosome from dad, and the SRY protein will influence this development. Here's the gender sign for male. And basically what you see here is that the genital tubercle will become the gland's penis. The urethral folds, if you look at them, they kind of fuse, and they form the ventral aspect of the penis. Now on the slide, I put the name of a muscle that's deep to the skin there, and it's called the bulbospongiosis muscle. Kind of wraps the penis at the root there, so I'll kind of put that in parentheses. That's a muscle you got to know. This bulbospongiosis, it's a muscle, and it's closed in the male. The labial scrotal swelling is a little outpouching for the gonads in the male. It becomes the labial scrotal swelling becomes the scrotum in males. The female gender is determined by the genotype XX, not XY, and that's kind of the default there. Well, anyways, just focusing on development. The genital tubercle becomes the glans clitoris. The urethral folds, they remain open if you look at the picture there. Okay? And they form the labia, basically. Labia minora. That's kind of a plural term referring to, labia means lips, there's two of them there, and there's that same bulbospongiosis muscle remains open in a space called the vestibule, and well, it's going to lead to the vaginal orifice, so it needs to remain open. And then the labia majora formed from the labial scrotal swelling. Now, if we look at an adult, this is a picture of a dissected view. I'll turn the lights off so you can see the red arrows a little better. And what you're looking at is, the muscles being indicated is the bulbospongiosis. So you can see here in the female, it remains open. That's the vaginal orifice. That's the urethral orifice there, external urethral orifice. In the male, the scrotal sac is reflected. There's the bulb of the penis, that's the muscle bulbal spongiosus muscle there. And that's the anus. Just to orient you there. So that's kind of one difference I want you to know, that muscle. It remains open for females and forms the bulb of the penis in the male. The other thing that's different about males and females is that the gonad remains in the pelvic cavity for females, but in males, it descends. Okay, at around 7-8 months, okay, certainly by birth, you should do a visual inspection or palpation to make sure that both testes have descended. So when a male is born, baby boy, the testes should be in the scrotum, but they don't begin there. As you can see from the picture, testes start the posterior pelvic wall. So I'm writing the testes, they descend, they start. Let's say on the, I'll just say posterior ab wall. Now, if you look at the top figure, the gonad, it's behind the parietal peritoneum, which is the lining of the body cavity. And the descent of the testes has to descend by itself into the scrotum through the inguinal canal. So I'm writing the testes descend into the scrotal sac through the inguinal canal. That's how they get it out of the body cavity. That's what the figure is showing you. Can't really see the inguinal canal, but we'll study it. A couple other things to note. The descent of the testes, it's not by itself. It's being pulled by the structure called the gubernaculum. Gubernaculum pulls testes down. And also know that as the testes is being pulled down, that it's behind the peritoneum. I pretend I'm behind a bed sheet, and I grab onto the bed sheet, and I'm being pulled down, and I take the bed sheet with me. It's kind of what it's doing to the parietal peritoneum. It pulls it all the way down into the scrotal sac. If you look at the bottom left picture there. So that pink double layer is peritoneum. Now it doesn't remain, a part of it pinches off. It remains on the anterior aspect of the testes, and it's called the Tunica Vaginalis. All right, so. So I'm writing the parietal peritoneum is pulled down with the testes. A portion pinches off, forming an anterior sheath or covering. A portion is pinched off. Forming an anterior sheath. covering the testes. And if you look at the pink covering, it's a double layer, but anyways, it's called the Tunica Vaginalis. So, you learned that men have a vagina. We're expecting that, I'm sure, but what does vagina mean? It means sheath, right? So in female vagina is the sheath for the penis during intercourse. But know that this Tunica Vaginalis is derived from, I'm underlining it, the parietal peritoneum. That's where it came from. Okay, let's move on. If you look at the sagittal section of the male reproductive system in an adult male, this is kind of what you see here, the big picture. And if you cut right down the middle, you're cutting in between the two gonads. So I put a testes epididymis as an inset. And if you look at an anterior view with the inguinal canal dissected, that's this picture right there. Okay. So I got a lot going on in this slide just to introduce male repro. And I've kind of boiled it down to 3 words. For the male role, let me back up a second. So humans reproduce sexually. You got the 2 genders. The biology is male, female. Okay? We teach both sexes. Sex is a biological construct. And I gotta do everything twice. I gotta teach the male anatomy and the female anatomy. They're very different. And I have to teach the male and the female physiology. And I boiled the male repro down to what's called the 3 E's. Erection, emission, ejaculation. So that's kind of the male contribution to humans reproducing. Summarize, go through it. So the 3 E's in male repro. Erection, emission, ejaculation. So if a male is to reproduce, the first thing is that you must achieve erection of the penis. It's the first thing, you take it one at a time. Specifically, it's an erection of what are called the erectile bodies. Basically, erection is a vasodilation event. Blood vessels that supply blood to the penis, they vasodilate. That allows blood to rush in. You engorge the erectile bodies with blood, and that's an erection. So I'm writing a vasodilation event. Blood rushes into the erectile bodies. Which become engorged with blood. That's an erection. So there are 2 different types. There's the corpus spongiosum and the corpus cavernosum. Let me write them down. Corpus spongiosum. There's one of them. And this kind of forms the ventral aspect of the penis. Okay, in the ventral aspect of the penis, there's something called the bulb and the glans penis. The other erectile body, and there's two of them, is the Corpus Cavernosum. So this pair of erectile bodies, they're dorsal. Okay, so they form the dorsal penis. At the root, attached to the pelvis, then they form these like two little, they diverge and form two legs called the crus of penis. So I'll just put crus. C-R-U-S means legs. The legs of the penis. Basically, the bulb and the crus, can't see it. It's the part that's deep to the skin attached to the pelvis. Basically half the penis you can't see. You got to do a dissection to see the whole thing. Now the pictures do a good job. I want to use the app to show you the erectile bodies. It does a little bit better job to point out in the pictures. Get this set up. All right, here we go. And switch it to this one. So here's the app. I just kind of have a preset. If you're using the app and you're like, man, I can't do that, it takes time. You can't remember if things are uploaded on YouTube, and you can take a look later. Let me see if you're awake. What did I highlight in green? Yeah, half of a testes. Okay, I'm just making sure you're awake there. I know you just took a test. So I highlight things in green. See, that's Tunica Albuginea. I don't want them to hide that. Corpus spongiosum. We'll do a multi-select. I'm going to isolate. So I've isolated just what I want you to see. The one corpus spongiosum. What you're looking at, approximately the parts that's attached to the pelvis, forming the root of the penis. That's the bulb. Okay, but it's corpus spongiosum. Erectile bodies, they're basically highly vascularized, smooth muscle. Okay, you go out distally, the ventral aspect of the penis. We got this is the glans penis. Okay. Now let's take a look at the Corpus Cavernosum. What I like about this app, if you put it back, you can do a fade others. So you can kind of see how it relates to other structures by fading them out. Oh, before I move on to the next one, I have a muscle highlighted, and that's called the bulbal spongiosis. Notice it wraps the penis. It wraps the bulb of the penis. It doesn't wrap the whole thing though, does it? It just wraps at the root, in this case, the bulb. Okay. So let's note that. That the bulbal spongiosis wraps the bulb of the penis. Write it down here. Doesn't wrap the whole corpus spongiosum, just at the bulb. Okay. Now let's look at the other one, Corpus Cavernosum. It's the dorsal aspect of the penis, so I'll get out of here. Let's go there. Multi-select. course, do the pelvis with it. Let's isolate. See what that looks like. So what I've isolated is just the paired corpus cavernosum with the pelvis, so you can see how they diverge proximally. And we wrote that down. What are these called? The legs, yeah, the crusts of the penis. Okay, so there's two of them. They form the dorsal penis. There's a muscle that covers the crust of the penis. It's called the ischio cavernosus because it covers part of the corpus cavernosum and it's attached partly to the ischial bone, that right there. And there's one on the other side. Right there. So let's write down the name of that muscle. Ischio cavernosus, I-S-C-H-I-O, ischio cavernosus muscle. So what? These aren't basically part of the penis, but they're muscles that wrap parts of the penis. Okay? And their action is to, when they contract during an erection, they help maintain the erection because it's like they're trapping blood in the penis. So they help maintain erection, basically. So that's the first thing. That's an erection. So you can achieve an erection, have intercourse. Then you must have emission. But before I move on, is there any questions on the erectile bodies? People already know some of this information. For example, what's the name of the cell that fertilizes the egg? Sperm. Right? That's important for a fertile male to reproduce. It's made in the gonad, and it must be moved to the urethra. That's emission. So the definition of emission is movement of sperm up to the urethra. Just to give you a quick overview of the structures sperm must swim through, there's an acronym called 7UP. The S is Seminiferous Tubule. You studied that. Where is the location? Inside. Yeah, and then in the testes, right? Okay, E is Epididymis. The V is Ductus Deferens. That's an important one. N is nothing. 7. Oh wait, no, I forgot an E. The second E is ejaculatory duct. And the N of 7 is nothing. UP is basically urethral penis. Now, I put a space there because the definition of emission is all movement up to the urethra. So you stop, boom, there. That's emission. All right. So let's look at that briefly on the app. Let's look there as we can. So that's cool. You can like, there's testes blowing up there. Let's hide that. You can look inside there. You can see seminiferous tubule lit up. Okay. Get out of there. Get to the epididymis, which is basically a highly convoluted little tube, lies over the testes. Epi means overlying. Didymis means twin. I guess you got two of them. Well, anyways, it's continuous with the vas deferens, now highlighted in green. I'm going to back up a little bit. So look at the green tube that I've highlighted. It's very important because to move out of the testes to get to the ejaculatory duct, you got to go out of the scrotal sac. And the structure is called the spermatic cord. You're going to go through the body cavity, through the inguinal canal, and you just kind of step back, way back. And do you see how on both sides the vas deferens is like going inside the body cavity? It's going to go over the bladder, okay, and it's going to go behind the bladder. Let me throw in bladder. Okay, let's go posterior now. There's a posterior view, and we can see the end of the vas deferens. This is still part of it. It's called the ampulla vas deferens. We'll punch out the details later. That's the prostate, just inferior to the bladder. Females don't have a prostate. So to get to the ejaculatory duct, this tube pierces the prostate, and it's called the ejaculatory duct. So let me hide half of the prostate. So you can see inside there, prostate, hide. Oops, that. Okay, there we go. So I'm zooming in on the half of the prostate there. The structure I've highlighted in green is the ejaculatory duct. It's inside the prostate. Okay, and this is the first part of the urethra. It's called the prostatic urethra because it's inside the prostate. So basically, movement of sperm up to here is the definition of emission. I'll turn the lights back on. So the definition of ejaculation is movement of sperm out of the urethra, through the penis. Sex arousal is a parasympathetic event. Ejaculation is a sympathetic event. There's an old saying I heard, it's called point and shoot, right? Point, shoot, parasympathetic, sympathetic, just to help you remember that. And also, the urethra is shared by urine and semen, but never at the same time. Another thing is, well, there's a lot of things that happen, but one of the muscles we wrote down, it helps provide the expulsive force for ejaculation. That's bulbal spongiosis. Bulbo spongiosis. Provides the expulsive force of ejaculation. So let's look at the urethra and the half picture I have. Turn the lights off. So there's a lateral view. Here's the urinary bladder. During ejaculation, there's a sphincter that tightens to keep urine in the bladder so it doesn't dribble with the semen there. That's the first part of the urethra in the male. It's in the prostate. That's the prostatic urethra. Then you have to get out of the body cavity, and there's a sheet of muscle, a diaphragm of muscle you have to get through. It's a small membrane. So the part that pinches through that membrane is called the membranous urethra. Now here's the bulb of the penis. The rest of it is basically inside the penis. And that's the corpus spongiosum, so it's called the spongy urethra or the penile urethra. There it is there. Let me pull back out. Kind of see the rest of it there. And that's the urethra, about 8 inches long in males. But that is ejaculation. Turn this off. So this is where we were in the PowerPoints. And I have labels on, labels off, and I have other pictures here. These are models like from the classroom there, because you have to get very familiar with those models for the lab practical. So we have models for you to study basically. And so I try to put in pictures of the models, like next to pictures of the Yoler Atlas, so you can see that both are useful to study. And one thing I want to take a look at that we don't look at in the females is the inguinal canal, because remember that the testes has to exit through there. And also, because it lives outside the body cavity, the testes need a lifeline called the spermatic cord. It needs blood supply. The vas deferens has to communicate with inside and outside the body cavity. It needs nerves, and it needs everything. Okay, and they have to go in and out of the inguinal canal. And we're not talking a very big space. It's basically in the groin area, and it's about this long. Okay. Basically, a canal long enough to go through the ab wall. So it's important to consider for the male. And the Atlas does a good job of showing students all the muscle layers. And if you remember your anatomy, the ab wall muscles are sheets of muscle, and there are 3 layers and that you have to go through. And so this one is the external abdominal oblique layer. Let's write this thing. So the inguinal canal, know the layers of ab wall that the spermatic cord has to go through. One layer, external abdominal oblique. There's an opening presented in this layer. If you look medially, connective tissues of this muscle layer, they kind of crisscross inferiorly. And there's an open medially called the superficial inguinal ring. That's how the spermatic cord basically gets out. Superficial inguinal ring. Basically, the opening for the spermatic cord. So know the opening. Also know the inguinal canal has a floor. It's a ligament you may have heard of, the inguinal ligament. It runs from the anterior superior iliac spine to the pubic tubercle. It's right there. Inguinal ligament. just wrote forms the floor. Another thing to note, as the testes kind of like gets pulled out and the spermatic cord is formed, coverings of the testes and spermatic cord are formed from these ab wall layers. It turns out connective tissue from the external abdominal oblique layer form what's called the external spermatic fascia. So I listed here because they form from the external ab oblique layer. Now, if you dissect the outermost layer away, you have the internal abdominal oblique that's deep to it. And so that's kind of what you see here. Here's the internal oblique. And then what you see is that muscle fibers from this layer are kind of being taken along with the spermatic cord. And they form what's called the cremaster muscle. And the full name is internal abdominal oblique. Have cremaster muscle. Now, there's something called the Cremasteric Reflex. If you bathe in cold water or wade in a cold pool, just cold weather, or sex arousal will make this reflex occur where the muscle fibers contract and they pull the testes close to the body cavity for warmth if it's too cold. The reason why the testes are there to begin with is because basically, well, they like a little bit cooler weather. It helps the spermatogenesis, but if it's too cold, this reflex happens and it pulls the testes closer. Okay, so that layer is derived from the internal abdominal oblique. The deepest ab wall layer is transversus abdominis. There's connective tissue called the transversalis fascia. And there's also the deepest opening called the deep inguinal range. You know that. Deep inguinal range. And there are connective tissues that form an internal spermatic fascia around the spermatic cord and the testes. Note that, internal spermatic fascia. So if it helps to remember tie ice, what does it refer to? We got an outline there. This goes from deep to superficial. What do you think the T stands for against the deep layer? Transversus abdominis, internal abdominal oblique, external abdominal oblique. And the ice are the scrotal layers that you have to know. I'll underline it in red. Going from deep to superficial, the I is the internal spermatic fascia. The C is the cremaster muscle layer, and then the E is the external spermatic fascia. That's what we want you to know. So these are coverings of the testes and spermatic cord, and I put another picture that kind of shows it all here. here what you're seeing is all three layers dissected in a way where you can kind of see them. You just window through the first layer. All three layers are shown. They even show you the spermatic cord cut open, and they show you all three layers. External spermatic fascia, the cremaster muscle fibers, and the internal spermatic fascia. Although it's not labeled, but you know, should know that. So I took a couple pictures of the model. Students always ask me on the model. Well, the inguinal canal would be basically from here to here. So this opening here and here would be the superficial inguinal ring. The deep inguinal ring will be the deep one here. Okay. Here's a superior view. That's the bladder. And you can see the vas deferens going over the bladder and posterior bladder. And if you cut the spermatic cord open, I just want you to know there are a few things there. And you can use this picture to study. What we see here, when you dissect the inguinal canal open, you can see the epididymis and testes, but in the spermatic cord, sperm during emission have to swim all the way up through there and go behind the bladder to get to the ejaculatory duct. So the most important content of the spermatic cord is the tube that carries the sperm. And that would be ductus deferens or vas deferens. Okay, so write down ductus deferens, about 18 inches long. It's the most important thing in the spermatic cord because this is a repro unit and it carries the sperm. Know the blood supply. There's the veins are called the pampiniform plexus. So if I point to anything blue inside the spermatic cord, it's a picture. It's not really blue, but pampiniform plexus blue. Well, you can also call them testicular veins. A little easier to remember. There's a lot of them. Plexus means like complex network. There's one, usually one testicular artery. Arteries distribute blood to the whatever organ it is. So maybe blood flow is down towards the testes. And arterial blood's hotter, and you want to cool that blood down. And so there's a reason why you have multiple veins. There's kind of a counter-current exchange of heat. So here's the veins. Veins run next to arteries usually, and blood is flowing in opposite directions. Veins are returning blood to the heart, the spent blood. They're collecting it, returning it to the heart to get oxygenated by the lungs. But anyway, blood is flowing the opposite way. The cooler venous blood is kind of taking the heat from the hot arterial blood, so the blood is cooler when it reaches the testes. That's why there's more testicular veins and arteries. That's why I call this a counter-current exchange of heat. That's what you got to be able to identify there, and just look at there's that one red tube, testicular artery, and there's many testicular veins. You can see that room there. We have this picture. Turn the lights off so you can see just a little better. If I point to anything red, say on a lab practical, what would you identify it as? Yeah, testicular artery or testicular vein. And see that beige color thing? What do you think that is? It is inside the spermatic cord, and it's called the Ductus Deferens. Now, what if I kind of put a piece of tape around this whole structure and say identify? What would you call it? The spermatic cord. And you should even be able to identify the layers of the spermatic cord. For example, a red color indicates muscle. What muscle fibers would these be? Cremaster. I realize you haven't started studying yet. You're just taking notes. It's okay. Well, deep to that would be the internal spermatic fascia. There's a lot of things that are shown on the model that are test you on. So I did a close-up picture of this model here. And I'm indicating that you have two layers here, cremaster, and it's not a complete layer, is it? One muscle fibers covering, and then the internal spermatic fascia. Now over here, they just cut through 3 layers, and you have to be very careful what you're looking at. It goes gray, red, gray, from outer to inner. So the 2 grays will be external, internal spermatic fascia, and the red layer in between is cremaster. Okay, so I just want to point that out to you. They are shown. Another way to study this is on other models. Here's a model we have, and they show the cremaster muscle layer very well. Okay, and they show this double layer anterior thing derived from the peritoneum, is the Tunica Vaginalis, right? We talked about that before. That's another layer. So we have that model in the room. We also study the cross-sectional view of the spermatic cord, and let's look at what we can identify. And it's easy to teach from this, but I will look at a histology slide that we have with the spermatic cord. Again, what did I say was the most important thing in this cord? The thing that carries the sperm, the Ductus Deferens. Right here, boom. Always look for that. And here it is on the histology slide. And I even see a little purple splotch of sperm inside a very muscular tube. Okay, the muscle helps move it in mission. I see one testicular artery. On the histology slide, it's the bigger blood vessel. And it has a thicker vessel wall. Right? And it's surrounded by a bunch of smaller ones for the countercurrent exchange of heat as the testicular veins. And there's other things too. There's the internal spermatic fascia, the cremaster muscle, which I see was kind of faded out, but it's there. So you can identify these things on the microscope as well as pictures. That will be expected of you. The next slide I have just kind of the image of the histology slide by itself with it labeled to help you study and look at it yourself. And it's kind of blurry because I really zoomed in on this whole thing is the Ductus Deferens. There's a lot of smooth muscle, and there's an epithelium, a columnar epithelium, with sperm inside. So let's kind of note that. Tag on to the Ductus Deferens. Lots of lots, not losts. Lots. Lots of smooth muscle. I'll describe the epithelium as a pseudostratified columnar ET with stereocilia. Okay, we're going to move on here. Let's go back down to the scrotum. And we have a couple of pictures that will take us through all the scrotal layers. It's not just skin and gonad. There's many layers in between, and we've already learned some of them. But I want to be very complete and make sure we kind of just list them all together here. We'll just start with the skin. Okay. The skin of the scrotum has a wrinkled appearance because the layer deep to it, tunica dartus, is a smooth muscle that can contract and give that skin a wrinkled appearance. So tunica dartus is a layer of smooth muscle. I like the picture. What they do is they put a pin between the skin and the tunica dartus, a whitish layer, just so you can see the two. Now, deep to that is our three layers that we talked about before that are derived from the ab wall. The external spermatic fascia, the cremaster, the internal spermatic fascia are all shown there. 1, 2, 3. External spermatic fascia, cremaster, internal spermatic fascia. If you go right on top of the testes, you have the layer that's derived from the peritoneum, tunica vaginalis. It's a double layer. So if you pull back one layer, that's the tunica vaginalis, the parietal part. The part that's right on the testes, giving a whitish appearance, is the visceral layer. It's a double layer. So you got tunica vaginalis, parietal layer. You have tunica vaginalis, visceral layer. If you cut the testes open, you can kind of even see another layer. Let's do that. So this is kind of just a dissection all the way down to the gonad. This is a picture where it actually shows you inside the gonad as well. And the layers that we just outlined are numbered here. So layer number 1 is skin. 2 is tunica dartus. Then it goes 3, 4, 5, just in order. External spermatic fascia, cremaster, internal spermatic fascia. Okay, then it goes 6A and 6B. So the 6A is the tunica vaginalis. Remember, the tunica vaginalis is not a complete covering. It only covers basically the front, because this is the layer that's derived from the peritoneum, so it's not completely enveloping it. So the 6B is the tunica vaginalis, the visceral layer. And then there's a layer deep to that. So I'm going to continue on over here. The deeper layer is Tunica Albuginea. Albino implies what color? White. It has a whitish color. And don't be confused. There's a Tunica Albuginea here in the testes. But there's also something called Tunica Albuginea of the penis. particularly, well, I'll just show you, you tell me. Let's go back and look at it. Hold on. Flashback. Here's the dorsal aspect of the penis. Remember the name of that erectile body? Corpus Cavernosum. You see this whitish membrane around it? That's also called the Tunica Albuginea. So I just don't want you to confuse it. There's penis and scrotum there, okay? There is both. Well, anyways, let's go back to the scrotal picture, scrotal layer figure. Because you can see that the Tunica Albuginea, there's these infoldings that form what are called septum. Tunica Albuginea, septum, and the septum create little apartments inside the testes, and they're basically called lobules, testicular lobules. So, infoldings of this layer form what are called septum on the figure. In the septum, they create these little partitions called lobules. Turn form testicular lobules. And it's the lobules that are containing the seminiferous tubules. Okay. So within a lobule, one lobule, may contain anywhere from 1 to 4 seminiferous tubules. Each tubule may be 32 inches long. And there's a lot of these lobules. Okay, I can't remember the number off the top of my head, but each tubule is 32 inches in length. So they're highly coiled up into these lobules. The tubules are. Basically, I'm trying to convey to you, there's a lot of tube. There's a lot of surface area to make developing sperm. So we got all the way down to the structure where the sperm are made, and that's us, as far as I'll go. Okay. We went from skin all the way to seminiferous tubule. If we take a macro view and just look at the testes and epididymis together, that's what you see. Okay. Know that the epididymis does have a head, body, and tail. In emission, to get out of the sperm, you go to the head first. There's little ductules that go to the head first. Write that down. Sperm swim, get to the epididymis, head first. You always want to put head of whatever you're talking about. Head is a descriptor, and you have to put what you're describing especially when you're filling in a blank on a practical. Epididymis. D-I-D-Y-M-I-S. Epididymis. Body, tail. Now it's hard to see, but it has a covering. If you were to dissect it, there's a highly convoluted tube. And if you were to stretch it out, I've read that the epididymis can be about 20 feet in length. 20 feet. That's pretty far. Pretty long, I should say. I remember in high school, I could broad jump 20 feet. But anyways, the point is, again, there's a lot of space, this is actually, think of it as a storage area. Store sperm here. And also, sperm can mature here. This is kind of where sperm develop their ability to swim. They develop their ability to whip their tail. Side sperm storage. I'll just say sperm develop local motion in the epididymis. So it's one tube. And the thing about it is, if you cut that tube, you see many cut tubes, but they're all one continuous tube. If you look at the picture on the right, it's the histology view. Identify cells inside. Sperm, yeah. And let's take kind of a closer up view. I believe this is a low mag. There's a high mag view. I want you to see the splotch of sperm in the middle, spermatocytes. This is a pseudostratified columnar epithelial tissue with stereocilia. Okay. Down. Pseudo stratified columnar ET epithelial tissue with stereocilia. I have epididymis, vas deferens side by side. They're both tubes, and how are you supposed to tell one from the other? Be able to recognize the smooth muscle of the vas deferens. That's the difference maker there. Because the cut tubes of the epididymis don't have a lot of smooth muscle. The vas deferens is a singular tube. It's got a lot. Single tube, lots of smooth muscle. You know, it looks like a lot of cut tubes, but if it's really one tube, and there's no smooth muscle there. That's kind of a histology objective there. So we did testes, epididymis, so the penis. We've been talking about it. The penis, this is the kind of the platform for the root of the penis. And we have two muscles here. I know this pelvis happens to be female because there's two holes. One's for the urethra and one's for the vagina. But it applies to males too. We have this area here, which is called, the region is called perineum. Just write that down. Perineum is the region between thigh and buttocks. Those small bicycle seats, they kind of fit in the perineal region. They kind of trace it out here. You kind of draw this. That's kind of the perineal region. That's kind of the shape of a bike seat. Well, this triangle is called the Urogenital Triangle, just as a space. Although they didn't draw it as a triangle, this is called the anal triangle. You could draw it as a triangle. Well, anyways, it contains the anal orifice. We're not concerned with that, but the urogenital triangle, if you dissect the region, there's a muscle that spans the width of the pubic arch. It's called the deep transverse perineal. There's also a thin band called the superficial transverse perineal. Those muscles you should know. So you got deep transverse perineal, as well as superficial transverse perineal. So note those two muscles, because the penis is basically sitting on top of it. The root of the penis shown on the left, right here, and you can see the deep transverse perineal muscle right there. So that's why we noted it. And again, I always give you page numbers for the 3rd edition of the atlas. Let's again mention the penis. After you dissect away the skin and the coverings, you have the 2 erectile bodies. They're attached. Here they're illustrated, separated. I did this dissection once. I had to use a scalpel to separate the glans penis from the corpus cavernosum. They are firmly attached. When you're looking like that, basically it looks like a hot dog in a bun. But you have the 2 erectile bodies, and note the muscles that cover them at the root. Okay. I was doing this dissection, these paramedic students were having their lab, and I was on the same cadaver. They probably thought I was weird. What's this guy doing with the penis? I wanted to see this. Okay. I wanted to see how the 2 erectile bodies are associated with each other. You know, this is what anatomists do. People might think we're weird. This is our job. I have to like figure this stuff out in a cadaver and present it to students. And you know what we see from the picture is that corpus spongiosum. I should mention, both erectile bodies get filled with blood, but only the corpus cavernosum kind of gets hard in an erection. The corpus spongiosum fills with blood but remains soft. And what we see in the corpus spongiosum, in the root of the penis, proximally it forms the bulb. Distally it forms the glans penis. At the root, at the bulb, distally forms glans penis. And know that at the root, it's covered with, you know, same name, bulb, bulbospongiosis. Corpus cavernosum, remember it's described as being paired and dorsal. And at the root, you have the crusts, which are covered with ischiocavernosus. So it's not the root, the part that's attached. The rest of it is just called the body of the penis. And so I have a couple of cross-sectional views of the penis at the root or the body. And the difference is, at the root, the erectile bodies are covered with their muscles. So here it is here. There is the ventral aspect of the penis. That's the corpus spongiosum, but it's the bulb. So that tube is the spongy urethra. Here is the paired corpus cavernosum. Here they are at the top there. That's the bottom there. So, if I had to identify this muscle covering the legs, what is it? That's the Ischio cavernosus. What about covering the bulb? Bulbal spongiosis. Very good. Now, there's no muscle there, right? Kind of call this the monkey face. Resembles that, or the mouth of the monkey. Well, anyways, that's the urethra, the spongy urethra. Surrounded by corpus spongiosum. Dorsally, you have paired corpus cavernosum. Now, there are veins there. I would note this one. The deep dorsal penile vein, that's shown on a lot of our muscles. Deep dorsal penile vein. Well, there's other ones there, but that one's on a lot of our models, so we'll be able to identify that. And well, those are the veins on top up here. When these become engorged with blood, these collapse. It helps prevent blood from leaving, basically trapping blood in the penis to help maintain an erection. Here's an histology slide next to the illustration. I'll turn off the lights. Can you see the monkey face? Well, you should be able to. I have it labeled. Right? There's one, there's the other. Corpus cavernosum. There is corpus spongiosum surrounding. The lumen is the lumen of the spongy urethra. If I zoom in on the epithelium that lines the urethra, it is a transitional epithelium. I want you to know that. That lines the urethra. So let's do that. So know that corpus spongiosum contains spongy urethra. It's got a transitional ET that lines it. Well, we can pick it up here next time, but before you leave, there's something I want to show you. I just posted it on Canvas while you're taking your test. It's called, well, it's basically the Lab Practical 1 study aid. I'll show you what it is. Spent a lot of time making it. It's new. I didn't do female yet, but to help you study for your Lab Practical, I have a lot of models that I test students on. And what I did was I took pictures of our models and I made a list, just all the stuff you got to know. I'll cover it all eventually. So for female, I'll do that next. But what I did was in taking the pictures, I just labeled everything. So I do one labeled, then unlabeled, but with the leader lines, and then blank. Okay, and I do that for everything that I think I could possibly ask you. These are all pictures that I took of our models in the classroom. And so I really want you to study these models using this document at home. Let me get to the male stuff, because I did that today. Here's male, there's a summary list. And there's labels on, labels off, and then blank. So you can study this at home. I don't have a lab scheduled until like Monday. But by Monday, I want you to have studied this. I want you to like know the stuff. know that the lab practical does cover 2 chapters. Let me jump forward to development when we get there. So development is the second chapter covered in this lab practical. So I basically just put all the models over there. I took pictures of everything. And I've taught this class enough to know all the questions you ask, and I labeled it accordingly, so that it's something you can study. And so those are pictures of models, but I also use pictures from the book. The students always ask me, "Oh, which one are you going to use?" And show you all my cards. Pictures from the book. I didn't label these. You have the book. I did for all the models. You don't have access to that. So to be fair to you, I labeled it. Okay. You have the book, so all the pictures. Yeah, I showed that one today. I showed that one. I showed that one. So I could use any of them. How it breaks down is your practical. It's about 80% models, about 20% pictures. But of all the pictures, I just gave you all the ones I could possibly use for both chapters. If you have to look at this document, it's a lot of slides. And, yeah. Class dismissed. I'll see you on Friday.